I'm Dr. Mary Pardee, and this is the Modern Wellness Podcast. Today, I'm joined by somebody who I met at an aging conference, Daniel Promislau, who received his doctorate at Oxford University in 1990. It was during that time that he developed an interest in the evolution of aging, including a focus on differences in lifespan across species. In 1995, he joined the Department of Genetics at the University of Georgia, moving to the University of Washington in 2013, and recently relocated to the USDA Human Nutrition Research Center on Aging at Tufts in Boston. Dr. Promislau studies aging and genetically variable populations. Just as individuals all look different, we also age in different ways. The challenge is to understand both how and why individuals differ in the patterns of aging. To explore these questions, Dr. Promislau uses a broad array of approaches: fly experiments, genetics, metabolomics, cross-specific comparative studies, computational and statistical methods, mathematical theory, and a wide range of species, from fruit flies and marmosets to companion dogs and humans. Dr. Promislau has been recognized with numerous awards, including the Ellison Medical Foundation Senior Scholar in Aging Award, a Breakthroughs in Gerontology Award, the Glenn Research Award from the Glenn Foundation for Medical Research, and the Bennett J. Cohen Award in Aging Research. In 2007, he was appointed a Fellow of the American Association for the Advancement of Science. Dr. Promislau is the principal investigator and co-director of the Dog Aging Project, a longitudinal study of tens of thousands of pet dogs throughout the United States. Together with his fellow Dog Aging Project co-founders, Dr. Promislau recently launched the Dog Aging Institute, a nonprofit institute dedicated to advancing innovative research into healthy aging in dogs to benefit dogs, their owners, and the veterinary community. All right, how are you doing today?
I'm doing great, and I'm happy to be here. Looking forward to our conversation.
I'm really, really excited to have you. We met at AGE, the conference this spring, all around geroscience and longevity, and I was a little out of my element for sure. I think I was one of three clinicians that were there. Most people were like you, researchers, PhDs, and there was a lot that was going over my head for sure, which was humbling. It was also very interesting for me. So I appreciate all of the stuff that you guys are doing.
I actually wish we had more clinicians there, and the field has evolved so much since I came into it, which is in the late 80s, hard to believe. And we're now at a point where so much of the work that people are doing is either translational that has the potential to get into clinic, and we actually are now seeing geroscience discoveries being tested in the clinic. And I think it's so important for the basic scientists and the clinicians to come together and work together to think deeply about all the implications of this new work that's happening.
Yeah, and that's how we started talking. We were just sitting at the same table, and I started picking your brain. But I think those conversations are so important because I love hearing from people who are actually doing the research. Hey, what are your thoughts about some of the things that are happening in clinical practice? And would you do it, kind of thing? And we're going to go through some of that today for sure. And separately, I'm realizing we may have overlapped at Tufts for a little bit. I was there in the Friedman School. I did a research internship in their energy metabolism lab
Oh,
in
cool.
Boston, which I think is the same. You're in the Friedman School, right? That building?
I'm not in the Freeman School, but it's just down the street. So
It's
I'm
down the
in
street.
it's called the Jean Mayer USDA Human Nutrition Research Center on Aging, a little long-winded, and it's just around the corner from the Friedman Schools. It's a center focused on nutrition and aging, but they're separate. I'm brand new here. I just started my full-time job in March of this year, so I'm not sure
Wow.
if we
We
actually
didn't overlap then. Yeah. I was 2010 and I was the person in the lab doing like bomb calorimetry and like bench side stuff. So, but great experience for sure.
yeah.
I want to jump in. How did you get involved with dogs?
So a few times in my life, I've done a major pivot in my research in, and it's been inspired by a paper that came out. And so in 2007, I was a professor at the University of Georgia at the time in Athens, Georgia, in the Department of Genetics, which was just a block away from the veterinary school at UGA. And this paper came out end of 2007 from the lab of Elaine Ostrander, who is one of the global leaders in dog genetics. And she's now at NIH. The paper was on the genetics of size in dogs. And this is such a great question because dogs are so variable. As anybody knows, you just go to the dog park or walk down the street and you're going to see Chihuahuas and Shih Tzus, and you're going to see Great Danes and Bouviers, and you might even see a Mastiff if you're lucky, 200-pound dog. So what's going on there? It's all genetics. The paper came out in Science, and on the cover of that issue was a picture of a Great Dane and a Chihuahua walking side by side. And it turns out, unlike in humans, where there are thousands of genes that probably impact size, none of which contribute more than a tiny, tiny little bit to the overall size, the variation that is in size. In dogs, there's a single gene that explains almost half the variation.
Wow.
That gene is insulin growth factor 1, or IGF-1. And that's what Nathan Sutter, who was the first author from Elaine's group, discovered. And I read that paper and I got excited because I knew a couple of things already. First of all, IGF-1 has long been thought of as a gene that's important in influencing aging.
Mhm.
And the second thing is that I've done my graduate work, my PhD work on aging in mammals across different species, and partly about trying to understand why it is that larger species tend to live longer than smaller species. Dogs are the opposite. The large breeds are shorter lived than the small breeds. So I put all this together and I thought, gee, I'd love to work on this question. My friend Steve Ostad, who's well known in the aging world, is married to a veterinarian, and she had told me earlier about a database of tens of thousands of dogs and when they die and how big they are. And all of this came together. This paper came out, but I knew nothing about dogs or veterinary science. I reached out to the vet school, and after getting the runaround, I get this email from Dr. Kate Creevy, who says, I don't know anything about aging and probably can't help you, but I'm willing to meet with you. And we met, and we've been working together ever since.
Wow, wow. It's so many things there. First of all, just that correlation. We all know Great Danes don't live as long as some of the smaller, like toy dogs, but that clicked with me when you're like, that's not the same in other species because we have like elephants, right? Versus a mouse. Those are going to be opposite. Out of curiosity, what is the longest lived animal versus the shortest lived? Do you have any idea?
We have some pretty good ideas. The longest lived animal is probably a clam. There, we have evidence for these clams that are quahogs, the same clams that end up in New England clam chowder, can live 500 years or more. So the next time you have a bowl of clam chowder, think
Wow.
twice. Greenland sharks, few hundred years. Whales can live hundreds of years. So not all the largest species. Some of the longest lived species are probably some trees that can live thousands of years.
Yeah.
But in terms of vertebrates, mostly larger species, also species that live in cold water. So maybe temperature has something to do with that. On the short end, many of your listeners will think of mice and voles, little rodents. They certainly live long, but not all rodents. So one of the shortest lived vertebrates that we know of is this little killifish from Africa called Notobranchius. And these little guys live their entire life cycle in less than 2 months.
Wow.
Uh, but the interesting thing is they can spend many months, they can spend a year in suspended animation as a fertilized egg in the dried mud of a dried up little pond in Africa. The rains fall, and right away they start developing, mature, and have the next generation in less than 2 months. And people are
Wow.
studying aging in those species in the lab. So we can learn a lot from species that live long like we do, but we can also learn a lot from studying the species that don't live as long. And the cool thing about dogs, which we're going to mostly talk about today, is that we kind of have both. We have breeds, they're all one species, but we've got breeds that are really pretty long lived, especially some of the toy breeds. And then we have some of the giant breeds that don't live very long at all.
Yeah, 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 that's really interesting. And the shorter lived species, I'm sure, are nice to study because you can get a longevity study done much faster than, I mean, that's why doing humans is so difficult.
Yeah, so we think about being able to complete a study within the lifetime of a graduate student. Of course, our graduate students, we hope they all live a long lifetime,
Yeah,
but
right.
within our labs, they're just there for 5 or 6 years. And typical grants from NIH are 5 years. And so we're always asking ourselves, what can we accomplish? How can we have deep insights into aging with studies that aren't going to last more than 5 years? Which kind of, jumping ahead, one of the huge challenges with the Dog Aging Project is that the insights that we're going to gain are going to take more than 5 years. And
Yeah.
so we have to structure those studies for the long term. And we can get to that later.
Yeah, yeah. So why dogs in terms of are they a good model for human aging? Because that's also, I know you want to know about dogs themselves, right? That's part of the aim of the study, but I'm guessing some of this is meant to be translated, hopefully, to humans.
A great question. There are a lot of reasons that dogs are good to study as dogs for dogs, and also, though, to your question, because of what they can teach us about humans. So, humans are hugely genetically variable. We're all different because of our genes as well as our environment. One of the challenges with all the species that we study in laboratories, mice, nematode worms, yeast, fruit flies, those are the big four, and some others like the killifish I mentioned. They tend to be highly inbred, so there's no genetic variation. We inbreed that genetic variation out of the populations we're studying to control for everything. We're always trying to control for all the possible variables except the one we're studying. So we remove the genetic variation. We keep them in a constant environment for the same reason. We live in a variable environment. You and I live in different environments. We're genetically different. And so that's a huge challenge if we're going to understand why we age differently, because all that variation in aging is due to genes and environment. So dogs are a great model, partly because they're genetically variable and their environments are as variable as our environments because they live in our environments. And in
Makes
fact,
sense.
when we go off to work, they stay home. So they're even more exposed to our environments than we are in many cases. Also, they get a lot of the same diseases that we do, diseases of aging, kidney disease, and loss of cognitive function, and cancer, and osteoarthritis. So many diseases of aging that we get, dogs get. There are some interesting exceptions, like cardiovascular disease. They
Mm,
don't have heart attacks, they don't get strokes. They have other heart problems, but they don't get those diseases that, of course, are major challenges for us as we age. And that actually is an opportunity for us to understand what protects them. So, for all those reasons, they're genetically variable, environmentally variable, they get the same diseases as we do. And then, importantly, they have this really sophisticated healthcare system with the same kinds of treatments, and including not only treatments for diseases, but preventative treatments as well. So, a huge opportunity for us to study a species that lives like us and does what we do as we age, but so much faster. It's not like a mouse or a killifish, but they don't live for 90 years. And so we can gain these insights in a much shorter period of time than it would take for human studies. There are
yeah.
these long-term human studies, but the dog gives us special opportunities to learn really important lessons that much more quickly.
Yeah, yeah, it's a nice in between from mouse to human and not quite because primates aren't living with us either, right? Like we don't have that advantage of having this similar environment. So, yeah, this whole new subset of variables that can be examined, I think is really powerful.
And there's enormous power in numbers too. A lot of what happens, especially when there's so much variation, to understand what the different elements are that are contributing to that variation, you need big numbers. Sometimes those contributors have small effects, and to identify those effects, you need big numbers.
Yes.
Sometimes diseases are rare, and so to identify the effects of particular diseases, you need big numbers. We have 48,000 dogs in the Dog Aging Project, and we're still recruiting, we're still growing. If any of your listeners have dogs, sign them up, dogagingproject.org. And it's hard you can't get those kinds of numbers in a study of non-human primates, for example.
Yeah, yeah, you guys aren't taking on a small project either. Like this is a monstrosity. So thank you for doing that. I know it's not easy.
Yeah.
Okay, let's dive into it. What is the Dog Aging Project? And I do want to circle back on something that you said, so I'm going to take a note, but if you can start there.
Sure thing. So the Dog Aging Project is a long-term longitudinal study of the genetic and environmental determinants of healthy aging in dogs. What are the genetic factors that shape aging? What are the environmental factors that shape aging in dogs? And these are dogs in people's homes. So these are the dogs that we live with, experiencing their regular daily life. We are just observing them. And then we do a kind of deep dive into the molecular biology to try and tease apart the mechanisms by which the genetic and environmental factors shape healthy aging. We have enrolled 48,000 dogs into the study, and we are continuing to enroll dogs every day. New dogs are welcome. We have dogs of all sizes, ages, and sexes. So there's no specific eligibility criteria other than the dog has to be in the United States. And we hope to someday expand to other countries as well. There's a small part of the project, about 1% of the dogs are enrolled in a clinical trial testing the ability of a drug called rapamycin to increase what we call health span and lifespan.
Yeah, and I want to dive into that separately too. So that's a subset of this 48,000, right? You have a group that is just doing
About 1%. Yeah.
1%. Okay. And then in these dogs, what are the main things that you are tracking and you're interested in right now?
I'll tell you, the reason I went into aging, I was a graduate student in the late 1980s, and that's when I started studying aging. And I thought it was just a temporary phase. And then I would, as a young student, I would get back to studying other things. And it just kept pulling me back because aging is like the great connector.
Mhm.
It connects everything in biology. And in fact, it connects everything, period. If I'm sitting on an airplane on my way to a meeting and I'm sitting next to someone who works in business or economics or is an artist, you name it, there's a way to connect aging.
Mm.
And so your question is such an important question. What are the specific questions we're interested in? And I will say, because we're studying aging, we're interested in everything
Everything,
because
yeah.
aging influences and is influenced by everything. But in particular, we're interested in asking what are the environmental factors and biological factors that help dogs live the longest, healthiest life possible? And what happens as a dog ages? In human medicine, we have fields like geriatrics, and a geriatrician can tell you if you are an older person yourself and go see a geriatrician, or if you bring an older friend or relative to a geriatrician, they will quickly be able to tell that person how well they are doing as an older person, what's going well and what maybe is not going as well as it could given their age, if it's 70 or 80 or 90. Veterinary medicine has many of the same specialties as human medicine, orthopedics and ophthalmology and oncology. Veterinary medicine doesn't have geriatrics.
Hmm.
And so veterinary students aren't routinely taught if a border terrier comes into your clinic, here's what to expect with age, and here's what a good ager looks like versus a Shih Tzu or an Affenpinscher. Now, for the common breeds like Golden Retrievers and Poodles and German Shepherd dogs, a veterinarian is quickly going to learn what the healthy agers look like because so many of those breeds come in. For the rarer breeds, not the case. So one of the things we're trying to do is to understand what we call normative aging. What does a healthy ager look like? What does a not so healthy ager look like? And what are the things that we can do? What might be within our control and what maybe is not in our control to predict who is going to become a healthy ager? What are the things we can do to increase healthy aging as dogs age?
That makes a lot of sense. Yeah, for the rarer breeds to be like, well, is this normal at this age for them to come in with this thing, whether it's like hair loss or rash or, whatever's going on. So fascinating. I don't know how veterinarians do it in terms of they have to know so many things about so many different breeds and animals. I'm like, humans
Yeah, my
alone
so
is more
my
than enough for me.
my one of my co-founders, Katie Creevy, likes to say to her students, she's a veterinarian, internal medicine specialist who teaches at Texas A&M, and she likes to say to her new students, when a dog comes into your clinic, the first thing you're going to do is genetic analysis with your eyes. You're going to look at that dog, and if you know the breed, right away, you're going to know what is the dog at risk of, what is it protected from, just because of what you know about its genetics.
Oh, interesting. Really interesting. I want to try to pull out of you. What are the biometrics or like the things that you're tracking that you're most excited about to get the information from that you think that we'll learn something that's applicable for humans?
There's so much, and we are a big team of about 100 people who are working directly on the Dog Aging Project. So I'll tell you what I'm interested in, but it's certainly not the only interesting thing. There's so many exciting things that people are working on. In my own work, I've always been interested in variation among species and genetic variation within species. It's really hard to tease apart the genetic contributions to variation. I hinted at this before. One of the challenges is that for many complex traits like aging or even something like body height, there are lots of genes that contribute, and each contributes a very small amount to that variation. What we've found is that if we lift up the biological hood of an organism and go inside and look at the molecular biology under that hood, sometimes that can help us link the genetics and the downstream traits. In my lab, we do something called metabolomics. We study the metabolome, which is a measure of all the metabolites. Those are the small molecules that are circulating in all of us, some of which your listeners will know, like glucose or amino acids or vitamins. These are metabolites that are circulating in us and dogs and almost all organisms. And when we measure those, we find that they help us kind of bridge the gap between genetics and our traits of interest like aging, and also point us to mechanism. And so we're just now analyzing some of these molecular traits. We're analyzing the metabolome. We have another colleague on the project who's looking at the microbiome, trying to understand all the microbes in our gut. Another colleague who's looking at what we call the epigenome, which is a measure of the not the actual sequence of genes, but the structure of genes. If you look at the genes in a nucleus in a cell, are they open and available to be expressed, or are they tightly closed and turned off? And that can tell us a lot about an organism, including its age. So right now, we're just writing up these papers on all the molecular biology, and I feel like it's this huge piece of the puzzle. And my hope is that we're going to discover biomarkers. These are things that we can measure that will help us predict the future of a dog. How likely is it to get a certain disease? How long do we think it's going to live? What are the best treatments if it has a particular disease? So the molecular biology and the study of these biomarkers, the hope is that they'll allow us to not only do better veterinary medicine for all dogs, but to do what we call precision medicine, where prevention and diagnosis and treatment is tailored to each individual. So we hope that what we learn from dogs will teach us about precision medicine in humans as well, so that when you go to your physician, he or she will be able to tell you not only whether you have a particular problem or not, but what you're at risk of and what you, as the individual person that you are, can do to make sure you live the healthiest possible and longest possible lifespan.
Yeah, yeah, that's definitely the goal. That's how I work with our patients too. But we also just don't know enough right now to be able to do a lot of that. So that's why this work is so important. And I'm just thinking when we're evaluating a human, if you're looking at things like bone density, a frailty index, you're looking at blood markers like CRP or ApoB or Lp(a) or cholesterol levels, right? Do we do something, or even like a MoCA, right, for mental health in terms of looking at dementia risk? I'm guessing we have very similar things in dogs. They just look a little bit different.
We do indeed. A lot of people on the project are working on many of those different aspects. Some of the really exciting work coming out of the project, and this is not my work, not my area of expertise at all, actually has to do with cognitive function in dogs. It turns out that very much like in people, as dogs get into their geriatric years, they are at risk of what we call canine cognitive dysfunction. It looks a lot like Alzheimer's disease.
Hmm.
It involves memory loss, loss of what we think of as executive function, knowing how to get out of a corner. So you'll see dogs that get lost under a table or in a corner and they don't know how to back out. They don't remember where their food is. They may not recognize their owner. We are studying the potential causes of that, asking if some of the causes or biomarkers of Alzheimer's show up in dogs with canine cognitive dysfunction or CCD. Some of our initial analysis suggests that dogs that exercise more tend to be at lower risk of CCD. We don't yet know if it really is predictive, and if so, how, but the nature of our study, that we're studying these dogs over the long term, will eventually allow us to answer that question. We also do many of the standard clinical analyses in dogs that we do for humans. So, for example, if you take your dog to the veterinarian and there's something wrong with the dog, there's a good chance that the veterinarian will have what we call a CBC Chem UA, or complete blood count, blood chemistry, and your analysis. And if you look at the CBC Chem UA from a dog that's done by a veterinary clinical reference lab, it looks very much like a human CBC Chem UA. The healthy reference values may look different, but very much similar. And then we're also doing some more sophisticated things. So for what we have 48,000 dogs that have enrolled. Not all are still alive, of course. Within that group, we have some sub-cohorts that we sample more intensively, and the most intensive of that is the precision cohort. In addition to getting blood for CBC Chem and the urine sample for urinalysis, we also do a lot of things like the metabolomics I mentioned in my lab. We send a blood sample to our collaborator, Ann Avery, who's part of the project at Colorado State. She's an expert in what we call flow cytometry. So she takes the blood cells and she runs them through the flow cytometer, and that tells her a tremendous amount of information about all the different kinds of immune cell types in a dog's blood. And we can infer a lot from that about the current health of the dog, as well as its previous exposure to infectious agents, for example. So there are all kinds of things that we are interested in doing. Many of the standard biomarkers in humans we don't yet have in dogs, just because we haven't yet done the work. But I'm excited about the potential of the Dog Aging Project to provide the veterinary community with more and newer biomarkers that will tell us about both diagnosis of current disease and risk of future disease.
Yeah, yeah, really interesting. I know I'm going off track here, but do you see insulin resistance in dogs being present?
So dogs are interesting, and I should say I'm not a veterinarian. Important for your listeners to know, I'm not a veterinarian, and I cannot nor will I give veterinary advice.
Totally.
We have seen, as your listeners know, a real epidemic, a global epidemic of metabolic disease in older individuals, obesity-related type 2 diabetes, a global epidemic. I mentioned already, dogs don't get cardiovascular disease.
Yeah.
They also don't get type 2 diabetes,
Okay.
but they do get type 1 diabetes. I don't know why it is exactly that they're protected from type 2 diabetes, although their cholesterol metabolism is different than human cholesterol metabolism.
Mhm.
So there are so many similarities, so many that we can study, and where I think the dogs will be a really powerful model for understanding and treating disease in humans. But there are some interesting differences that are also opportunities for study, but also where we need to be cautious about saying the dog does everything that humans do. There are important differences.
Yeah, yeah. I mean, especially given how heart disease is the number one cause of death in terms of humans with aging. So
That's right. What we do see in dogs is heart failure, and that's obviously really important. So as humans get older, they are greater risk not only of cardiovascular disease, heart attack, myocardial infarction, but also heart failure. And we see heart failure in dogs. We see mitral valve disease in dogs. So there are some similarities
Yeah.
in terms of the challenges that old dogs and old people face with heart function.
Right, 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 right. And what do we know about dog size and patterns of disease and aging if we haven't covered that piece yet?
One of the really interesting things about dogs is this variation. Go to a dog park and look around. There's so much variation in what they look like, how they're behaving. The same variation exists in disease risk. And a lot of that is associated with size. Not all of it. There are some breeds that are at unique risk. For example, we have a curly coat retriever, and he's a carrier for a gene that causes a metabolic disease if you have two copies of the mutation, and the curly coat retriever is the only breed known to get this disease. But size is really important, and there are many diseases that are common only in small breed dogs or only in large breed dogs. And there are important diseases in humans as well. Certain kinds of cancer. So dogs are at high risk of cancer, but generally, the larger breed dogs get more cancers, especially of certain kinds. And there are some important differences. So many of the cancers that unfortunately we see quite commonly in pediatric cancer patients, kids, but are very rare in adults, like osteosarcoma, for example, bone cancer. We see that in some kids. There are certain breeds that are at very high risk of osteosarcoma, and they tend to be large breeds. Disc disease, many of us have experienced herniated discs, for example, intervertebral disc disease. Some of the smaller breeds, especially the sort of longer, shorter breeds, are at risk of disc disease. Osteoarthritis is common in dogs. Kidney disease, common in dogs. We talked already about neurodegenerative disease, cognitive disease. So size is associated with cancer. We see a lot of that in the larger breeds. Heart failure, we tend to see in the smaller breeds. So it's interesting. There are some diseases that go up with size, where large size is a risk factor, others where large size seems to be a protective factor. So it's interesting. We see going in both directions. It suggests an opportunity for us to discover causes where we find
Yeah.
these correlations. That's what excites me about this issue of size. It's kind of a window into what we might be able to do to understand at least some of the reason that diseases occur in some dogs and not in others.
Yeah, yeah. As we're talking, I'm realizing this study is logistically really complex, right? It's not like you have men and women and they're in the same age group and that's kind of your variables there. It's like you have different breeds, sizes, sex. I'm sure you have neutered, not neutered, or they all have to be neutered.
Oh, we have everything. This is a great comment that you've made. And really, I think it's important to think about logistics in two ways. First of all, there's the logistics of creating this study. And I mentioned that there are about 100 people on our team. They're not all scientists and veterinarians. A lot of those people are people who are just brilliant at managing the logistics of creating and running a study like this. We have an incredible team of people who spend every day, all day long, figuring this out, answering phone calls and emails that we get every day from participants who are trying to figure out what they're supposed to do. People writing computer code that sends messages out. People figuring out the logistics of shipping samples so they arrive at the right place at the right time. So there's that, which I just have to give a massive shout out to
Huge.
everybody on our team all across the country who do this every day, without which the Dog Aging Project wouldn't exist. And then logistically, all of the different kinds of data types. I actually think of this as a kind of superpower of the study because it's so large and so variable in the way that you alluded to. It allows us to tease apart all the different elements that interact. Often, when you do a study in humans, you might find that two things are correlated, but years later, you'll discover they're correlated because both of them are influenced by some third variable that you didn't have available at the time. So we're trying to make sure that we have all the variables. We can't measure everything, but we try and measure as much as we can. The neutering piece is really interesting. I actually have someone in my lab right now who's been looking at some of these data. In general, so when people talk about sex in dogs, when veterinarians talk about sex in dogs, they actually talk about four sexes:
Right.
males and females, intact or sterilized. And so there are four possible combinations. It turns out from work that we've done in the past on previous data sets and work we're doing now on the Dog Aging Project data set, that sex is important. Whether a dog is sterilized or not is probably even more important in terms of predicting what diseases it's going to get and how long it lives. In general, dogs that are sterilized live longer than dogs that are intact.
Interesting.
Importantly, this is not a reason for people to go out and choose to sterilize their dogs.
Yeah.
That's an important decision that each of your listeners who has a dog or a cat should make with their veterinarian who's an expert in these areas.
Yes.
And I will say, for the same reason, there are long-lived breeds and short-lived breeds. The right breed for each of you is not the breed that lives long. For many people, the right breed is going to be a short-lived breed, and it's absolutely the right choice.
Yeah.
I just want to say to the sterilization issue. So in general, sterilized dogs live longer. Interestingly, they die of different things on average. So sterilized dogs are more likely to die of cancer and autoimmune diseases. Intact dogs are more likely to die of infectious diseases and trauma. But in general, the sterilized dogs live longer. So it's very interesting, and we don't know why. Something that we hope to tease apart in the coming years.
Yeah. Any insights on frailty or dementia in with those two things?
Not yet. So the exciting thing is we have the data. We are developing a frailty metric. So in humans, we have ways of measuring frailty where we combine lots of information. My colleague Kate Creevy, who I mentioned before at Texas A&M, is working on taking the existing data that we have on owner-reported mobility and these sorts of things and putting together a frailty metric. And then we are certainly going to ask, how does that change with age? How does that change with sex? A really important question that you're asking, because of course, as you know well, as people age, women live longer than men. In fact, females live longer than, have higher survival than males at every age, starting from conception. Even in utero, female fetuses have a higher survival rate than males. But as people get old, frailty in females is greater than in males. We're talking about people in their 70s, 80s. So their survival rates are higher, but their frailty is also higher. And we don't know why. And we're interested in whether the dog model might help us understand and answer that question in humans.
Yeah, yeah. And I'm guessing dogs have estrogen, progesterone, testosterone, right? We call them slightly different things and they look a little bit different. So that's a really interesting piece. At the conference that we were both at, that was a big part of what we were discussing is sex differences and how underrepresented women are in general in research. And this project brings a whole nother element to it because you have both sexes and sterilized and intact. So to be able to view what may be the effect of these hormones and what happens when you take them away earlier in life. And I'm sure it depends when too, right? But so many interesting things are to come from that.
Yeah, and something I'll add, which is really a new field, and we have some papers we're just writing now, we haven't even yet submitted them, beginning to think about the idea of the effect of sex not simply being binary. There's not just a male effect and a female effect, but the effects of being a male or a female might be modified by our genetic variation.
Mhm.
And in dogs, the majority of the dogs in our study, about 90% of the dogs are sterilized. Most people in this country sterilize their dogs. It's different in other countries. One of the interesting things about that is that removes a lot of the hormonal differences, not all. We already are seeing that in some of the molecular biology that we're doing, a lack of difference between males and females that are sterilized, but it also creates an opportunity potentially to better understand how genes modify the impact. So, for example, we know that there are some diseases of aging that are more common in men than women, and others that are more common in women than men. But that doesn't mean only women get them or only men get them. The question is, might that also depend on your genetic background? We don't know the answer, but we now think we might have a good model to begin asking these new questions.
Yeah, interesting. So it's almost acting as a way to partially control for certain variables to be able to see what's actually at play there. That's fascinating.
That's right. Yeah. So sex is a major impact factor, but then the genome, we can think of it as kind of a rheostat, tuning the effect of being male or female.
Yeah, fascinating. I want to turn gears a little bit because a lot of my listeners are interested in human longevity. And so we're going to start to play in between these two and just talk about rapamycin in general. First, what does it do? Where did it start slash it still is used in human medicine? And why has it been brought into the longevity space at all?
Yeah, it's a fascinating story. It's a long story that I will make much shorter, and I'll preface this by saying that so as I mentioned earlier, about 1% of the dogs in the Dog Aging Project are involved in a study called TRIAD. That stands for Test of Rapamycin in Aging Dogs, TRIAD. It was initiated by Matt Kaeberlein and is now co-led by a team of Kate Creevy, May Reed, Roz Anderson. Rapamycin goes back decades. It was discovered in a soil sample from Easter Island. Easter Island is also called Rapa Nui. So Rapa Nui, rapamycin. It ended up in a freezer in Montreal, and someone took it out and started looking for drugs that could inhibit fungal growth. And it turned out that rapamycin was seen to inhibit fungal growth. And that led someone to ask some years later whether it might inhibit. So fungi are not humans, of course, and not mammals, but someone thought, well, maybe if it inhibits fungal growth, can it inhibit cancer cell growth? And sure enough, it did. And long story short, that ended up leading to rapamycin being used in human therapy, but it's not mostly used directly to treat tumors, for example, but in transplant therapy. So both in solid organ transplant patients and in bone marrow transplant patients, rapamycin is given because it attenuates a pathway involved in growth called the mTOR pathway. And it turns out in solid organ transplant patients, it prevents rejection of the organ, which is a foreign body brought into the patient. And in bone marrow transplant patients, it helps prevents rejection by the transplant because all those immune cells now that can do rejection come from a donor into the patient. So there are thousands of people all the time around the world getting rapamycin to help prevent rejection. And there are ongoing trials in cancer patients. Eventually, people asked whether at low doses, because it's attenuating this growth nutrient sensing pathway, mTOR, whether it might help aging. And studies were done in mice that showed that at lower doses, much lower than human patients get, rapamycin could increase lifespan. And in fact, in mice, it improves as mice age. It reduces the risk of heart problems, cognitive problems, musculoskeletal problems, in addition to making them live longer. How it does this is still an open question. Some people believe that it is a mimic for caloric restriction. And we see many of these same benefits from caloric restriction in mice. If you ask Matt Kaeberlein, who initiated our project and who is one of the real experts in the field of rapamycin and aging in general, he will say it may turn out to have something to do with reducing the degree of inflammation. So it does a lot of different things. And it's a really interesting time. There are now, have been and continue to be clinical trials in humans. Of course, the challenge is there's no way to do a clinical trial in a human asking whether rapamycin will make people live longer because it would just take many, many decades.
Yeah.
But we can do that in dogs because the lifespan of a dog is that much shorter. And that's what we're doing. I've said a lot, but I might have lost track of the specific question that you
No,
were
no,
asking.
you actually hit them all, and it's a big topic. It honestly should be its own podcast, just this one thing. So I appreciate you condensing it. And out of curiosity, how are you dosing the dogs with rapamycin? Is it per kilogram of body weight adjusted for what most people are using clinically for humans, or?
Yeah, so it's a milligram per kilogram per week. So they take the dose once a week. And we've set up the study as a double masked placebo controlled study. So that means that half the dogs are on a placebo pill that looks just like the regular pill, the other half are on the regular pill. The dogs have to be a minimum weight and the minimum age. The reason for that is we don't want to study dogs that are extremely likely to live for another 10 or 15 years because the study will never end.
Yeah.
We want to study dogs that are a reasonably high risk of mortality so that we have the statistical power to see if we're reducing that risk. If we study a group of dogs that have a really low risk of mortality, high risk of survival, we just won't, we'll never see an effect. We'll have to wait too long.
Yeah.
And we're going to treat the dogs for a year, but then we follow them for an additional 2 years. So the study is actually a 3-year study, and we're constantly enrolling dogs. We are continuing to enroll dogs so that the dogs have to be middle-aged or older. They have to be 40 pounds or larger. We have about 20 sites around the country. So if people are interested, there's likely to be a site near them. The dogs have to be healthy and they have to be sterilized. And the reason for that is that there's some evidence from mice that rapamycin reduces fertility. Interestingly, a little bit of a segue here, there's an ongoing study by Yushin Sa at Columbia looking at the ability of low-dose rapamycin in perimenopausal women to increase the number of years that they will maintain fertility. And the study is still in the early phases, but the idea is that treatment with rapamycin reduces the number of follicles that are shed every cycle, the number of eggs that are produced, and so leaves more eggs to be produced later. We don't want to impact fertility in dogs, and that's why we're using sterilized dogs.
Yeah. Yeah. That's interesting though. And so they're doing a human clinical trial.
So this is a human clinical trial with low dose rapamycin, low enough that don't appear to be any side effects that we see at the higher doses for transplant patients, focused just on fertility. So it's a relatively short-term trial, not a trial of lifespan. And Joan Manick published a few studies in recent years treating older individuals with rapamycin and asking whether it would improve immunity response to vaccines, and seeing some compelling results from her work as well.
Yeah, 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 really fascinating. I asked you this at the conference, and I always like to ask people who are deep in the field their personal thoughts. And it's like, do you take rapamycin? Because there's a handful of people, right, that are taking rapamycin right now, despite us not knowing everything about it and what it does and what it doesn't do and possible side effects. But curious to know your thoughts on that if you want to share.
So I'm not taking rapamycin. I don't take any supplements other than a vitamin D pill that my dermatologist told me that I should take every day. So that's all I take. And as I mentioned in our conversation at Madison at the age meeting, we have really, really good epidemiological data, tons of papers out there showing that health span and lifespan are improved by some interventions that we all can do. We can exercise, we can ensure a healthy diet, we can do our best to have good sleep hygiene. Of course, some people struggle more with sleep than others, even if they have good sleep hygiene. We can limit our intake of alcohol and eliminate any intake of tobacco products, and we can try to have a healthy social life. The epidemiological data point to all of those factors being associated with good health span and lifespan. And I try and do those things to the best of my ability. So those are my interventions. There are certainly people out there who are self-prescribing drugs that in laboratory studies have been associated with improvements in health span or lifespan. When I say health span, I mean the duration of lifespan that an animal maintains, during which an animal maintains good health.
Yeah. Quality
And
of life.
yeah, that's right. And rapamycin is not the only drug. There are many that people are pretty excited about. Some of the big ones out there, metformin, acarbo, 7-estradiol, there are others. One of the challenges is that we don't have clinical trials in humans showing that these things increase healthy lifespan with minimal or no side effects. And in many cases, we don't have any clinical trials on health, on aging related traits. As we discussed already, there are trials out there. I mentioned the work that Yushin Sa and her collaborator are doing at Columbia. There are many, many trials that people are doing on rapamycin and senolytic drugs to eliminate senescent cells and metformin and so on. But it's early days. And my training is in evolutionary biology. I'm a great believer in trade-offs. One of the things that you learn in evolution is that really there's no such thing as a free lunch. And I think we need to really be circumspect about whether there might be some negative consequences to these things that we hope will have beneficial effects. For all those reasons, I think it's early days. I'm very cautious about whether these things will improve health span in humans. And the last thing I'll mention, going back to one of our earlier points of discussion, we know a ton. We've learned so much about aging, really important lessons about aging from worms, yeast, flies, and mice in the lab under highly genetically and environmentally controlled conditions, which neither of which reflect our reality.
Yeah.
We have a paper that will be coming out in a number of weeks in Aging Cell showing huge, huge variation in how different genotypes of flies respond to rapamycin. Now, we're measuring development time, we're not measuring lifespan, but the variation is massive. There's some genotypes that don't respond to the drug, even when we increase the concentration 500-fold, and they still don't respond. And we don't know whether a drug at a dose that works for me might not only not work for you, it might even be deleterious, harmful for you. But we have so much more to learn before I think we can say this drug is going to help people live healthier longer. The last thing I'll say is, that said, I think we do have drugs that are well tested, that people are taking who are at particular risk, like statins, that are helping people live healthier longer. They're not appropriate for everybody. And of course, anyone who's thinking about statins needs to talk to their physician before they do anything else. I don't take statins. I don't have the risk factors for that. But where we have a lot of data, we're already at a case where we've discovered drugs that can help people live healthier longer. And that's a great thing.
Yeah, yeah. Yeah, I need to send this to some of my patients because there's such a kind of controversy around statins, especially people that are interested in alternative medicine and health, that they want to avoid everything. And sometimes when I'm talking to them, I actually talk like that, where I'm like, this could be a longevity drug for you. If you're interested in longevity, you could think of it like that, not like you're broken and we got to fix you kind of thing, but this could help you live longer. So I like that mindset shift because you're right. We do have good information that if we can help reduce the risk for heart disease in that manner, then it can be really powerful.
Yeah, again, I'm not a physician nor a veterinarian, so don't take any advice from me about that.
So you're still enrolling. How do people actually enroll their dogs in this project? And we can put links for this in the show notes for sure.
If you're interested in enrolling in the Dog Aging Project, go to dogagingproject.org and you'll see a link, and all you have to do is follow that link. You'll create a password protected portal and fill out a survey. Takes an hour or two to fill out the survey, and then your dog will become a member of the Dog Aging Project.
Yeah, and it's like a community thing that this project is becoming because of how much information we're going to get from it. And people love dogs. It's like dogs and babies, right? Like people love their kids and they love their dogs just as much, sometimes more maybe.
Yeah.
And
One of the wonderful things about the Dog Aging Project is that it brings people together from all walks of life. We ask a few questions about the participants, which they can choose not to answer, but we ask about things like the zip code and address of the home, which we don't share with anybody. We ask educational levels and income levels, and all those data are not shared, but they show that we bring together people from all walks of life, different income levels, education levels. We don't ask about politics, but for sure we've got people across the political spectrum. And that's so important that dogs bring everybody together because of their shared interest in dogs.
So important.
I just want to share one more thing, which we're really excited about, that my co-founders, Matt Kaberlein and Kate Creevy, and I also just launched a new initiative, which is the Dog Aging Institute. It's a 501c3 nonprofit institute. And we think of the Dog Aging Project as one part of a big umbrella of projects, some of which already exist that we haven't had time to talk about, and some of which don't yet exist, which are going to all be about understanding and studying healthy aging in dogs and people, educational initiatives. The Dog Aging Institute is designed to support all of those things. So we launched this 501c3 to bring in partners, to welcome donations, to support to help us support educational and research initiatives in this space, to bring people together to have these really important conversations. And I'm just excited to share that with your listenership.
Thank you so much for what you guys are doing. You're making a huge, huge difference in the geroscience community at large and just the human community at large. So we're really excited. I have one more question. You can do yes or no, because I know you got to run. There's a lot of people in the biohacking space that say, I'm going to live to 180 years old. They're in their 40s or 50s saying this. Do you think that that's realistic?
I do a lot of demography, look at the data a lot, and I think the data support a pretty strong limit. I don't think we're likely to get past the 120 roughly. Jeanne Calment is the one person who made it past that out of billions. And I think if the biohackers and biologists, whoever, can figure out ways to keep us healthier longer, that's great. As we get older and older, other problems I think are going to start to show themselves in the same way that we see a lot of Alzheimer's now that we didn't see 200 years ago. So personally, I don't think it's likely. I certainly hope that we can live the healthiest lifespans possible, no matter how long we live.
Yeah, great point. Thank you so much for your time today, Daniel. This was really insightful and appreciate you.
Thank you. Been a pleasure.
Thanks.
Bye-bye.
Bye.